thank you very much for the nice introduction, and thank you very much again for having me here. My name is Ali, a senior scientist that's working in the Constructor University that recently rebranding from the Jacobs University. So it was previously was famous as Jacobs, but now is rebranding to the Constructor University. Our group actually, and our group focus actually is on the network analysis, and it's led by Professor Mark Sturzen-Hood. It's the chair of computational systems biology in Constructor University. And actually we work on the interface of the statistical physics and systems medicine and systems biology. Actually, our investigation covers omics data analysis, omics data interpretation in biology and medicine, and the application of the network sciences on the medicine and biology. In general, we work on the different aspects of the application of the network on medicine, like drug repurposing, discovery of the disease gene and the driver genes, plus disease subtyping and disease association, how the diseases are connected to each other, and comorbidity data analysis. More specifically on the metabolomics data analysis, we developed different statistical modeling approaches for the different purposes. Batch effect correction is one of our tasks that is make us busy here, and multi-omics data analysis as well, and data integration, plus model construction and microbiome data analysis. This is whole package that actually is our routine task in Professor Hood's group. So already we heard a lot about the metabolomics, and I suppose you agree with me that it's really hard to summarize all metabolomics in a nutshell. But we know that there are some standard pipelines for the metabolomics data analysis, and I try to split this whole standard pipeline into three different parts, like data acquisition, data analysis, and data interpretation. And we already know that how sensitive each step would be. So like which instrument actually you use and which data format you get data from. And the technique that coupled with the mass spec also matters. So we have plenty of the different mass spec approaches like gas chromatography, LCMS, even imaging metabolomics also should be taken consideration. So in addition to that, the pre-processing approaches and data analysis has a big impact on the final result, and downstream data analysis like biological interpretation and pathway data analysis, plus correlation of or making correlation network out of this whole data. In addition to mass spec and the different derivatives of mass spec, there is one certain approach for measuring the metabolic flux of each reaction, metabolic reaction in the cell. We call it metabolic flux analysis. It has own challenges in data analysis. Why? Because samplings is different with the other techniques, and we need to label our compound and the substrate to find out how they follow through the network or through the pathway, and we can measure the fluxes of each reaction plus the rate of them. But technically it's not easy, it's not really straightforward. But it can quantify the fluxes of each reaction, and the principle is this technique is based on the mass balance, that the rate of the production and the rate of the consumption should be equal. And for the different purposes, we should use of the different isotopic labeling, like we use of C13 in some cases, or nitrogen 14, and conclusively, I can say that, we develop different pipelines for the data analysis, but all of them are sensitive. Somehow they are not reproducible. And most important is the data availability, that you cannot easily find the metabolomics data in the databases. For your study, if you are working on the cancer or the different metabolic diseases, non-alcoholic fat liver, for example, whatever, or some organelle-based diseases like Gaucher that is dependent of the lysosome deficiency or whatever. So the data is not publicly available as transcriptome and proteome. So we use of, let's say, surrogate for the MFA to estimate the fluxes out of the network. And briefly, if I can tell you about the different metabolic networks modeling, we actually have 3 different sets of the metabolic network modeling that start with the so abstract one we called network modeling or metabolic network as a graph. We consider whole metabolism as a graph, and we just consider the node that could be metabolite an enzyme. So we use of 2 different set of entity, the metabolic network, and we construct the network, and we run the different analysis on the network to find out, for example, the most important node as it could be as a drug target or biomarker, or we look for the motive. There are some motifs that are responsible for the stability of the network, so we can find out which network are more stable, and other analysis like clustering or whatever. So not more data or information we can get out of the network analysis. From one side, so it's one abstract and one extreme approach for the metabolism analysis, metabolic network analysis, actually. From one side or another extreme of the metabolic network analysis, we have a kinetic modeling. In kinetic modeling, despite of the network analysis that we just should know about the node and interaction, in the dynamical analysis and kinetic modeling, we need a lot of data, like exact value of the concentration or the inhibitor, activator, how enzyme works, the kinetic law of each enzyme, for example, and whatever. So this is not something that experimentalists like. So they should prepare a lot of data information and provide it. And from the computational side is highly computational demand. So these both extreme are not interest, they are interest of the scientific work, but not a lot. But in the middle, we have a constraint-based modeling, or let's say genome-scale metabolic model that usually we use of the flux balance analysis to estimate the flux out of the whole network. So briefly, you can see the differences between these 3 different approaches, that from the left to right, you see the difference in the size of the network. For example, for the network analysis or genome-scale metabolic model, we can use of the larger scale, but in the dynamical analysis, we just focus on the smaller scale because it's highly computational demand. But the result of the last one, the right one, is so accurate, but as I said, it's so hard to provide the data for that. So we go for the middle one that somehow takes some good characteristics of the left side and good characteristics from the right side, combine them in a genome-scale metabolic model and use them. So, but on top of that, the assumption that constraint-based model is based on that is the system is under a steady state. It's the rate of the consumption and production would be equal, and metabolic don't change in time. So briefly, if I want to show you how this system works, the core of the genome-scale metabolic model is this matrix. Actually, we call it a stoichiometry matrix, that on the top, on the column, you see all the reactions that contribute to the system that we are working on. And on the row, you see the metabolite, and the cells are occupied with the numbers that could be 0, minus, or plus. So minus means something is consuming, plus is something produced. And so this is the whole core of the genome-scale metabolic model. But in addition to this stoichiometry matrix, we have a vector of the flux that assign to corresponding reactions. So in this modeling, we have reactions, metabolite, plus the vector of the flux that assign to the reaction. And this flux can be changed in the different condition. So you can use it for the perturbation analysis or whatever. We will see later in the next slide about the application of the genome-scale metabolic model. So this stoichiometry matrix plus the vectors can shape can shape a solution space. All the answer that we are looking for is inside of the solution space. So how we can use of the solution space? It's depend of the investigation that you are doing, but in most cases we use of the biomass as an objective function to optimize the objective function to see how cell grows. And if this function turns to 0, it means that the cell is dead. But somehow we can find a point that indicates the optimization of the objective function on this solution space. Okay, so this scaffold actually the stoichiometry matrix plus the vector of the fluxes that form a solution space can be used as a scaffold for integrating additional information to the solution space. What do I mean? I mean that you can customize this solution space or this predefined scaffold to whatever you want. So from one side, we have all the reaction, transporter, all the compartment, organelle, whatever that we have in the organisms in the case of human. But in a bacteria or in a biotechnology, so you have the organism of the bacteria or whatever, you have all the reaction, and plus the whole reaction, you can use of the additional information like transcriptome, proteome, or even metabolomics data to integrate with this model and make it customized. So it could be tissue specific, it could be disease specific, cancer specific, whatever, and then you can estimate flux out of this scaffold. So it could be a really good surrogate for the FMA. So instead of the use of this all the metabolic approaches or metabolic data, we can easily use of this approach to estimate the flux. Of course, we need to validate at the end, validate our result, but it could be a good approach along with all the experimental data that we are producing. There are a lot of technical detail in genome-scale metabolic model. Just briefly, I can tell you some that, for example, GPR is one of the fundamental things that we need to have. So in most of the models is already done, and in some cases they are getting updated because of the new information are adding. But it's something super essential for the data integration. Why? What does it mean, GPR? GPR is gene-protein reaction association, actually. So it's worked as a Boolean law. So some genes have an AND as a Boolean law, and some genes have OR. What does it mean? If there are AND between some genes, it means that actually they work together, and if one of them knocked down, the whole reaction would be knocked down and deactive. And if there is an OR, it means that the two genes are like is an enzyme that if one of them is knocked down or deactivated, it doesn't matter. Other one can metabolize the metabolite that is there. So for the data integration, actually we need at the first step the GPR of any organism that we are working with. But there are also other technical details that which omic data is compatible with this framework, or which platform of the computational platform, that we can use, or which algorithm for the data integration is suitable for us, which functionality we can use, which phenotype, and how we can data integrate. These are all the technical details in genome-scale metabolic model that you should care about that. In nutshell, I can say now, in nutshell, we can say that it's compatible with almost all of the omics data from the transcript from different platforms, microarray, RNA-seq, even single-cell transcriptome. It's compatible with the genome-scale metabolic model. Proteomics data is also compatible with the genome-scale metabolic model and metabolomics. So feel free to use, but any of them has a disadvantage and advantage, and you should be carefully use of them for the data integration. Another technical details about the functionality of this models. So the first one and the most important one is gene essentiality. When you develop a model and integrate your data in the model, you should first check gene essentiality, that the essential genes that are relevant to this phenotype exist in your model or not, and plenty of the different metabolic functionality that are available, like you can use for the personalized medicine, drug target prediction, biomarker identification, even drug side effect for the controllability analysis, microbiome analysis, and metabolite optimization. Sorry. Which software and which platform are mostly used? Because from the last decade, most of the algorithms are developed in MATLAB. And one of the main toolbox of the MATLAB is Cobra for this type of analysis. Mostly people use of MATLAB for the genome-scale metabolic model, but there are also some package in R that you can use, like Sibyl, or in Python is also CobraPy. It's available if you want to use of Python and R. But all functionality of the Cobra is not developed yet in the R and Python. Just briefly, I can give you some impression how this integration algorithm works. This is just two different categories to show you that some of the algorithm works as a reduction algorithm. What does it mean? It means that when you apply this algorithm on your network, according to the additional data like transcriptome or proteome, it would shrink the solution space. So it could exclude some of the reaction of your model. But in some algorithm, they also care about the objective function. For example, let's say this algorithm are objective function-based integration approach. If you exclude some interaction or reaction from your model and the objective function turns to zero, it's taken back to your model. So it should be, this exclusion should be consistent with the objective function calculation. But some of them works with the one gene set. For example, you can see here GIME is one of the famous algorithms in this area. So just work with one set of the data, but we also have some algorithms that work with the several set of the data. And some of them doesn't work with the reduction idea. They just normalize the gene expression as what it is and integrate the whole them and diminish the flux according to the normalization aspect of the transcriptome data, for example. But there are plenty of these different algorithms. If you are interested in the integration algorithm of gene expression and proteomics to the genome-scale metabolic model, this paper could help you. That in this paper, we summarize almost all available algorithms and discuss about the advantage and disadvantage of algorithm, the code if it's available, and brief description about each algorithm. And about the application. I listen to all the talk and I really enjoy, and I can tell you for all this talk that I had, genome-scale metabolic model is applicable. Some of them I have some example here to show you, but for some I just mentioned that afterwards that they are also doable. For example, in this paper, we actually apply a new idea for the data integration. So we know all about the downstream analysis of the whole pipeline, that we are looking for some pathway, for example, which pathway is down-regulated, which one is up-regulated. But we got this idea and take the downstream analysis to the first pre-processing step. What I mean, so in data integration, actually, we integrate one gene to one protein to one interaction or reaction. But our idea was, first, let's do some enrichment analysis and then integrate the whole pathway on the model. So we care about enrichment analysis as a pre-processing step, not as a downstream analysis. So in this case, we apply this idea to the different scenario of the yeast metabolism, and we show that it's outperformed in some scenario to could predict better the metabolic behavior of the yeast by this approach. So there is still open room for developing of a new algorithm for the data integration. This is the last one, as I know, that we published this year. But in the optimization, we can use, for example, this is another study that we use of the Genome-scale Metabolic model to optimize some secondary metabolite, in this case, antibiotic. That is really important. We got Salehzadeh organism that's the source of the antibiotic production. And just by manipulating the medium, we try to optimize the antibiotic production that at the end, we validated by experimentalists that if we change, for example, the phosphor depletion we expose to the medium, at the end, the antibiotic production would be amplified. Today, we discuss a lot about the microbiome. So genome-scale metabolic model is also compatible with the microbiome data analysis. We can make, let's say, community modeling to let the microbiome interact to each other in silico and talk to each other in silico. And at the end, we can find out the whole behavior of the community model and the microbiome model to see how they show the differences in the different community. So, for example, in this study, we show that how microbiome in the different stage from healthy to adenocarcinoma, how the behavior of the microbiome metabolism changed when the colorectal cancer progressed. And we targeted some metabolite that in the later stage amplified in the colorectal cancer patients, like TMA, 3-methylamine, that we showed that is amplified in the later stage, or reduction of the butyrate. It was something else that we discovered in this study. But plus this, we showed that at the later stage, microbiome can promote inflammation, for example, by producing a lot of H2S or NO, for example. For sure, this study needs to be validated, but I believe that could be a hint for the experimentalists to just follow our finding that in reality can see some kind of changes in a real patient or not. So I told you this is still open room for if you are thinking about a new idea of to develop a new algorithm, or even there are not a lot of web-based tool for the data integration in the genome-scale metabolic model. There are plenty of the databases that you can use, like BIWG, that actually collected all developed organisms, genome-scale metabolic of the organisms. You can go through that, or virtual human metabolite that is specific for the human being, and the Recon3 that is lately released. So these are all available as a database, but for the web-based tool for the data analysis, I didn't see anything, to the best of my knowledge. So one of the study that I just want to go to the details is about the heterogeneity of the metabolism in cancer that we use of the genome-scale metabolic model to see how patterns of the metabolism are different within the cancer. And can we distinguish the cancer by the metabolic pattern, or if they are different, can we find out the most influential reaction that can distinguish between the different cancer or not? So we all know about the Warburg effect that goes back to the last century, actually, that German scientists discovered that the metabolism in cancer cell are completely different with the normal cell or differentiated tissue. So as I depicted here, you can see that the normal cell differentiated tissue, in the presence or absence of the oxygen, they show actually two different behavior. In the presence of the glucose, it's metabolized to the pyruvate and then enter to mitochondria and produce ATP. So for each glucose, we produce 36 almost ATP, but in the absence of the oxygen, we have another fate that it produces lactate at the end and 2 mole of the ATP for each mole of the glucose. But in the cancer cell, it doesn't matter. In the presence or absence of the oxygen, the behavior should be same, and glucose turn to lactate with the almost 4 mole of the ATP for each mole of the glucose. So it was the first metabolic hallmark of the cancer that Warburg showed in 1930-something and got the Nobel Prize for that. But so far, there are different hallmarks of the cancer that we know about that, like they reduce the pH of the microenvironment, or hypoxia, or redox stress, or whatever. But the question that we had to determine the Warburg effect results with the same metabolic pattern, or is driven by the different mechanism. Just in short, we find out that each cancer has its own fingerprint of the metabolism. So we can use of the pattern of the metabolism as a fingerprint to identify a cancer. And we can distinguish the cancer by this is the interesting part, that we found out the most influential reactions that we can distinguish between the cancer, or maybe it could be related to the drug sensitivity, is the peptide metabolism. Peptide metabolism, as we found out, are completely different in the different cancer. So, conclusively, genome-scale metabolic model is a really valuable tool for personalized medicine, for drug target identification, for organelle-based diseases, host pathogen investigation, immunometabolism study, and many companies so far founded a single department for the genome-scale metabolic model, like Novo Nordisk, Syntev, GSK, Sartorius. All of them has their own department for the genome-scale metabolic model. Sorry if it takes long. Sorry. Thank you very much for your attention.